Okay. Good afternoon. First of all, thanks to Pamjean for the invitation for this very interesting meeting on a very broad area of topics. And what I will do now is change gears quite a bit from what we have heard so far today, because what we'll do is we'll move to a completely different type of proteins called the nuclear receptors. So we're moving away from the cell membrane, we're moving away from kinases that float around in the cytoplasm, and we're going into the nucleus to a class of proteins that can directly activate genes. They're really transcription factors. I'll tell you a bit about, we've heard a lot about the kinome arrays. I'll tell you about another application that has been developed here, and that we've also helped to develop the next step, that you can use in this. Yeah, it's a different kind of peptide technology, essentially. So what we're talking about is this family of proteins, the nuclear receptor family. And what they have in common, is there a pointer? Somewhere? Yeah. Great. And what they have in common is this general structure. They all have a DNA binding domain, as you should have if you want to be a transcription factor. And they also have here a ligand binding domain, and we'll come back to that later in quite a bit of detail. And if you look at the 48 genes in the human genome that encode nuclear receptors, you can see that that there's probably one or more of your favorite proteins, or proteins that you've come across in different through different reasons. There's some classical endocrine receptors like estrogen and the progesterone glucocorticoid receptors here, but also retinoic acid receptors, thyroid hormone, vitamin D, so really classical endocrine receptors. What we have here in the middle, and that's what I will mainly be talking about today, is a class of nuclear receptors that are called adopted orphan receptors, and that comes from the fact that, if you look here on the right, you have these orphan receptors, meaning that they are true members of the family, but they haven't found their ligand yet, so they're really very pitiful and sad. But some of them have found a ligand, and that's why we call them adopted orphans. And the one I'll mainly be talking about today here is one PPAR gamma that is sitting right here with its two close related family members, alpha and beta. Now, how do these proteins function essentially? Well, there's two ways in which these slightly different ways in which they operate. And I will only be talking about gene activation in this respect, so they also repress a lot of genes, but we'll not be talking about that today. Some of these receptors reside in the cytoplasm. The ligands just find their way to the receptor, and then the activated receptor detaches from heat shock protein complexes and travels to the nucleus, where it can bind directly to target genes and activate them. Alternatively, there's also a class of receptors that already reside in the nucleus and often already on DNA, actually. Again, the ligands just find their way through the plasma membrane, cytoplasm, and hit the nucleus, where they bind to the receptor. So, in that respect, hardly any signal transduction required. Why have these proteins caught a lot of attention? It's because they're drug targets, and that of course, is what we've heard a lot about today. And I've just given you a few examples here, because there's much more to say about this, but some of the best-known examples are anti-androgens that are used in prostate cancer, anti-estrogens in breast cancer, that actually maybe tamoxifen is maybe the best-known example in this case. But I will also be focusing again on a PPAR gamma, where we have this class of drugs called thiazolidine diones, TZDs for short, that are used to treat insulin resistance. So nuclear receptors really are part of what is sometimes referred to as a druggable genome, together with, of course, a lot of other cellular proteins. Well, why has this PPAR gamma caught so much attention? Well, I think it has to do... Here. I think it has to do with what we all know is happening at the moment, is that we are suffering from a real obesity epidemic. It's actually... Often people say that it's just happening in westernized countries or in Western countries. Well, that's not the case at all. It happens all over the globe. And for example, Saudi Arabia is really an area of worry and also South America. So please bear in mind when people tell you that this is a Western problem, it's not. Here's just a brief picture of Europe, because we were used to seeing these slides from the US, but also in Europe, of course, you see that in general, all over Europe, obesity trends are going up. And why is this an issue? Well, it has to do, of course, obesity has to do with adipose tissue, with your fat tissue, and fat tissue is really at the center of a lot of human diseases, either directly or indirectly. As mentioned, there are these drugs, and that will be important for the rest of the talk, these to fight insulin resistance, which is like the pre-stage of type 2 diabetes. Some of these drugs indeed target adipose tissue and PPAR gamma in that, but there's a lot of diseases that are also linked to adipose tissue, including cancer, for example, atherosclerosis, obviously, and so forth. Already in what is it now, almost a couple of decades ago already, people realized that this class of drugs, these thiazolidinediones that had been used in the clinic already, but no one really knew what their cellular target was. And then in 95, people realized that these insulin sensitizers, so they make you again more sensitive to insulin, and thereby prevent you from developing full-blown diabetes, that these drugs in fact target one of these nuclear receptors, PPAR gamma. So these are really PPAR gamma ligands. And if you look at that small subfamily of PPARs, you see that they're quite alike in terms of homology. What is really striking is that PPAR gamma comes in two flavors here, gamma 1 and gamma 2, but the gamma 2 is really specific for adipose tissue, for white adipose tissue. So that maybe explain why hitting this protein gives you some kind of really fat-specific effect, because you're targeting PPAR gamma in the fat tissue, and that gives you all the improvements. In a way, it's a bit of a funny receptor, so to say. It's really promiscuous. If you look at, I don't know if it's visible in the back, but if you look at the different structures of synthetic and natural ligands that have been proposed for PPAR gamma, you see that structurally they quite diverge, which means that the ligand binding pocket that is sitting, of course, in this ligand binding domain is quite wide and open, and there's a lot of molecules that actually fit in there. But still, again, I mean, it doesn't mean that it's not a suitable drug target. It's just something that you have to bear in mind. PPAR gamma was already known. Of course, the protein is not there, I mean, just to be a drug target. It has, of course, a very important physiological function. And just to give you, it really is now regarded as the master regulator of fat tissue, of adipose tissue. And just to give you a couple of clues why that is the case, when you differentiate to become a fat cell, that's a very obviously, like any differentiation process, it's a very complex process. It's a whole cascade of transcriptional events happening with many transcription factors and also, of course, extracellular signals feeding into that. But it all boils down to one thing. The final step is upregulation of PPAR gamma. You need enough of that protein, and then you will become a true fat cell. And you can kind of recapitulate that if you just introduce PPAR gamma into a fibroblast, and the cell will become a fat cell. And it tells you that this is really the final step and the crucial step. Why is that the case? Well, if you look at what is downstream of this transcription factor, you get like a fat cell signature. You get all the genes, all the gene products that are really relevant for being a fat cell. So the proteins you need for import of lipids, for storage of lipids, also for what is called lipolysis. So if you, in times of fasting, for example, where you need to liberate energy again and it has to go into the bloodstream, also those genes, of course, are very fat cell specific, or quite a few of them. Many of them are controlled directly by PPAR gamma, so that's why it's having such a central role in this whole system. And finally, we now realize, in the last 10 years or so, we've realized that fat tissue is in fact not as boring as we thought it was, but it communicates with just about every other organ in your body, with your muscle, liver, brain, and so forth. And it does this through so-called adipocytokines or adipokines, just hormones, really fat hormones, and some of them are also under direct control of PPAR gamma. So that kind of explains why it's such a central player. And also there's evidence, of course, from mice, and this is just one example of a study like that. If you don't have this is in fact a fat-specific deletion, that should be, of course, of PPAR gamma. Fat depots are very small, and you see that many of the fat markers are down, so that again confirms that. That doesn't quite tell you maybe yet why these TZDs are so actually work, why hitting this molecule, hitting this receptor would actually be beneficial. Well, the idea is in a way, in detail, is quite complex, but just to give you the take-home message, it has to do with keeping fat where it belongs. What one of the big issues is in insulin resistance and subsequent diabetes is that your body, when you're being obese or being overweight and subsequently obese, is that your fat is being stored not in your adipose tissue anymore. You've reached the end of the capacity there, but fat starts being stored in your muscle and your liver, and that's where the problem, where one of the major problems is. What TZDs do is essentially, based on what I just mentioned, is two things. It will enlarge your adipose depot, because if you super activate PPAR gamma with these drugs, you will get more adipocytes. You will drive the differentiation process, obviously. So you will also become heavier as a side effect. The other thing is, of course, that you will upregulate all these fat-specific genes in the already and new existing adipocytes, and also in new ones, of course. So in general, you increase your storage capacity, and then you will get fluxes of lipids moving out from your muscle and your liver and going back into your fat again. And that is what makes you healthier metabolically. Well, that all sounds nice and dandy, but are these TZDs really the wonder drugs? Well, obviously not, because otherwise we wouldn't still be working on them. We had the whole problem solved. Thing is that they're associated with some serious side effects. As mentioned, you get modest weight gain, right? Because you increase your adipose depot, you get more fat tissue, which of course gives you more storage capacity and makes you metabolically more healthy, but maybe that's not what you want if you start treating an obese patient, right? That's not quite what they expect from you. The other thing is, and that's maybe more serious, is that it leads to fluid retention. This is a fact that is linked to the liver, to the kidney, and from that you might end up with heart failure. There's a higher risk of bone fractures. I don't think we know exactly how that happens. There's some indications, but we don't quite know why. But there's also a higher risk of myocardial infarction. I have to say that this is controversial, but it was 3 or 4 papers, I don't quite remember, published in a New England Journal, and a lot of comments with it. The shares of the companies plummeted overnight. It was really amazing that it happened, and there's a lot of criticism on the studies, but still. The net effect was that the AME banned these drugs completely, and the FDA banned them except from certain special cases where you can still use them. So it tells you what bad press can do for you, I think. So where do we stand now? PPAR gamma is an important regulator of adipogenesis, so the differentiation process and adipocyte function. And it seems also by hitting this protein, you can have you're actually achieving what you want in a way, but there's too many side effects maybe. So can we go on and do this in a smarter way? And there's many ways of doing this. Of course, you could also go for completely different drug targets, but if you're a bit stubborn and if you want to keep this up, then how should you do this? And now we'll move back to more of the molecular business, PPAR as a drug target and how this works molecularly. So as mentioned, it is a ligand-inducible transcription factor, and that is a short sentence, but it implies a lot. If you look at the ligand binding domain, this is only the ligand binding domain, and you see this whole spaghetti of alpha helices that don't make much sense to you. But what you can see is when you feed it a ligand, like here in yellow, that the structure changes quite dramatically. And if you look just at this helix, what is called helix 12, it folds now back over the structure of the protein. So, and that tells you something, of course, that there is quite a dramatic conformational change. But what happens as well is that you used to have here in pink an interaction surface in the unliganded state that was there. which you lose completely, but you create a new one here. You create a new interaction surface with these pink helices. And that is actually very relevant, because what that allows you to do as a nuclear receptor is to switch from an inactive to an active state. Because if you change your structure so much and you create these new interaction surfaces, what happens is that you can switch from so-called co-repressor complexes sitting on the receptor. The ligand hits the receptor. These co-repressors fall off because they need the surface that was originally there. Well, that is, of course, is disrupted, so the co-repressors fall off, and the co-activators come in hitting this new interaction surface that was created by the conformational change. So, in that respect, nuclear receptors really are what sometimes is referred to as molecular switches, right? You can really switch them on and off. These cofactors, a couple of remarks about them, and then we'll go into some data. They're a class of proteins, as mentioned, I didn't mention it yet, but they don't bind DNA themselves. And that's also why they're called cofactors and not transcription factors, but they bind the transcription factors. And they, for example, what they can do is that they bridge your transcription factor with the general transcription machinery, thereby stabilize your RNA polymerase II and keep the gene firing. Also, in the case of nuclear receptors, they often bind in a ligand-dependent way. And when you have antagonists, so anti-hormones, they will not bind because you will not get that proper conformation for the interaction. Some of these co-activators, or cofactors, I should say, harbor enzymatic activity. And that may explain why they're so important for the transcription process. But there is a large class of them. Maybe this is still an underestimation, but I would say at least 200. And of course, if you want to check them one by one, where do you end up with? I mean, that's a very difficult exercise and a very time-consuming exercise, obviously. So can we solve this in some way? And of course, yes, we can. If you look here at the co-repressor and if you look here at the co-activator, here on the right, what is really striking, and that was discovered already quite a while ago, is that the co-repressors, and the same is in a way true for the co-activators, both require only a short alpha helical motif to bind to this surface on the receptor. It doesn't look like much, because what you see here, maybe I should just depict this one, the LXXLL, so leucine, any amino acid, leucine, leucine. It doesn't sound like much, but still, I mean, that together with the alpha helical conformation is really what is critical for the interaction. And that means that you can use this information, that you don't need a full protein, but only a peptide. Then you can start thinking about peptide arrays, obviously. And the way to do it then, it looks a bit like this, is that if you immobilize peptides like that on a carrier material, here you have your ligand binding domain, and we often use the GST versions that can be recognized by fluorescent antibodies. In the presence of a ligand, it will bind to the co-activator motifs. It gets the right conformation, it will bind to the motif. And if you don't have a ligand, nothing will happen, essentially. It will not interact, you will not see a signal. And that, of course, is then held by the principle of the technology that is also, I think, there essentially in the kinome setting, that you have this wedge sitting beneath your array, and that anything that is unbound through the pumping mechanism will end up below, where you will not see it. So here you have your cofactor peptides in red, here you have your nuclear receptors in blue. When they don't bind, when they don't have ligands, they will just fall through, and when they do find ligand, they will bind here, and you will get a bright spot, known to many of you, of course, from the kinome setting. So in a way, this is not a kinome array, this is an interactome array. And to show you that this is actually true, just two examples. This was the first generation, so this is like 48 or 50 different cofactor peptides. What you see, for example, this is a co-repressor peptide, so it should bind in the absence of ligand. This is one in red, and when you see that the signal is gone after you've given it a ligand, and here is one in green. It's a co-activator peptide. It will not bind to the receptor, only if the receptor is in the right liganded conformation, and then you will get a bright spot, also depicted here in the diagrams. And that works very nicely. You can expand this kind of trick, of course. I mean, that is really what we did a lot in the beginning, just to convince ourselves that this is really meaningful. So we performed a lot of dose response curves, and what you see here, if you just give ligand, you see the co-repressors falling off, so the interaction with the co-repressor peptide being diminished, while the co-activators actually increase with increasing amounts of ligand. You get these kind of profiles. You can depict them in many different ways. This is one of the ways you can do this with these spiky patterns. Here again, the co-repressor that drops dramatically, and here a co-activator that increases quite dramatically. Just to give you an idea of what you can do with this and how this has really helped us in some areas of research, I will only touch upon one small bit of drug development, because we're not a drug development group at all, but we have tested some of these weaker ligands to see how they would behave in this kind of assay. And this is one of these weaker ligands, telmisartan, maybe known to you from completely different areas, but it's also a weak ligand for PPAR gamma. You see that it causes only weak interaction between one of these cofactor peptides, or maybe a little bit more here. And we could confirm many of these things in independent assays, so in this case with the GST pull-down assay. And again, that really gave us a lot of confidence that the approach is really meaningful. You can also use this, of course, the array consists of peptides that we kind of devised, right? I mean, based on literature, on known cofactors, so it's not a random library at all. But still, it doesn't mean the peptide library was based on information coming from from all 48 nuclear receptors, if you want, and what we're looking at often in these cases is just one. So that means that there are still new data points to be discovered there. And when we did this for PPARs, we realized that there was one peptide that was binding here, or actually there were a couple of here, all unknown interactions, not published before by anyone else, no link to PPAR or PPAR associated processes whatsoever. We decided to zoom in on one of them, referred to as TRP3. And as you can see, it's really a nightmare. These proteins often come under 6-7 different names. But anyway, it was TRP3. It contains one of these alpha helical motifs. That's what you, of course, would. That's of course where the peptide came from. That was on the array, so that's here. It's very well conserved between species. You get a decent dose-response curve with ligand. You get a ligand-dependent interaction. When you mutate the motif, the interaction is gone, so that's all fine. It doesn't tell you yet if it's biologically relevant, so we did take the next step. We checked it in this adipocyte differentiation process, in these kind of assays. You see that TRP3 is increased in differentiation, and more importantly, when you do the knockdown, differentiation is completely blocked. So it's not just an interactor, but it is probably an important interactor. We still don't know. It's such a small protein. It doesn't have any enzymatic activity. We don't know in which complex it is, but it's an important player. Yes, and then you can also use this approach, because as mentioned, you can use this essentially to test drugs. If you want, you can use it as a discovery tool to find new co-regulators, or at least ones that have not been associated with your favorite receptors so far. You could also use it in maybe a more fundamental setting, and I really believe that the combination of, I'm in a translational session, but still I think the combination between fundamental and translation is absolutely essential. One cannot do without the other. And I was also very pleased, of course, that the rare diseases were again mentioned this morning by Irene Nostad. We do a bit of work on one of these rare diseases. It's called familial partial lipodystrophy type 3. And lipodystrophies means that you have a problem with your fat tissue, essentially, as a human. So this is a human syndrome, and what these people look like, this is just a drawing of a woman with that, and to be honest, we often discover them in women, and maybe it's clear why. What we see is that these patients have very little fat in their extremities, in their arms and legs, and particularly no fat in their buttocks, while the trunk and the neck and the face is all normal. So that's why it's a partial lipodystrophy. You lack fat in some areas of your body and not others. And this is, again, I think the reason why we often discover this in women, because if you would find a man, maybe even with a belly, but with muscular arms, you wouldn't think that's a patient, essentially, right? Anyway. Mutations, this syndrome has been linked to PPAR gamma for quite a while. And mutations that are there are all heterozygous, so you have one intact allele, the other one is mutated, and they really seem to hit always, as you can imagine, the business end of the molecule. They either hit the DNA binding domain, and of course if you have a transcription factor that cannot bind DNA, you have a problem, or they hit the ligand binding domain. And there's a lot of things happening in this ligand binding domain, apart from ligand binding itself. As mentioned, you have this whole cofactor interaction surface there, so there might be funny things happening there. And it's also necessary for dimerization with its dimerization partner. So there's many things happening there. And the striking thing is that with these people, apart from having this peculiar appearance, they have massive insulin resistance or type D diabetes. They have dyslipidemia. They almost look like a very obese person that is suffering from all the obesity-related issue. And I think it has to do with what I mentioned in the beginning. If you become obese, you have a problem with your expansion of your fat depots. Here, of course, if you don't have fat depots, you also have the same problem. So you will start storing your fat in your liver and in your muscle and so forth. And then you will have many of the same features. Okay. We were interested in some of these mutations. Some of them really came from literature, importantly by the lab of Stephen O'Reilly in Cambridge, but also by others. But we're also currently finding our own mutations, so to speak. How do they behave in this kind of assay? And if we compare, like here, I mean, and forget about all the details, if you compare just the interaction profile of the wild type, which is here in black, and maybe you can just about see the gray bar sitting here, which is one of the mutants. Essentially, the bottom line is that if you test these mutants in a cellular context, or whether you test them on an NR chip, we'll get very similar information, essentially. And that, of course, was very reassuring for us. So with your wild type, you see a lot of changes happening, all here in black. And with some mutants that are partially defective, you get a partial profile. And with some that are completely dead, you end up with nothing, essentially. Or you get full retention of your co-repressors in this case. So that kind of fitted for us, so that we were very happy with that. And this was, of course, looking a little bit in hindsight, because what we did here is that these were known mutations. We knew their behavior in a cellular context, and actually some of them have also been tested in an animal context, and they confirm what we know from the human setting. But you can also do it the other way around. So nowadays, whenever we hit upon a new mutation, what we do is that we put the PAM chip a little bit up front, because we think it gives you some nice information right at the beginning. And just as one example, and that is learning us a lot about how this receptor works. It doesn't tell us much more about the disease, obviously, right? We know that the syndrome is caused by these mutations. That's not our area of interest. The way is to go for the natural mutations, because if you're hitting an amino acid in a natural setting, obviously, yeah, our simplistic view is that you're hitting something important function or functions. So here's a new one we found, the E379K. As you can immediately see, often these natural mutations are quite dramatic, right? You see a switch in charge, or here leucine to proline. Yeah, you can also imagine that will do something. These are really no exception. But to talk about the E379K, it's in the ligand binding domain. So one of the things we did is do a PAM chip, and really it's a bit weak here, but really nothing happened. And that was a bit surprising to us, because we really expected a defect there. And also, if we generated a cellular setting, a reporter setting, where we just focus on the LBD, nothing really happens. So again, a nice confirmation between the chip data and cellular data. So we think that although this mutation is lying here in the LBD, it's somehow talking with other domains of the receptor, or maybe talking with its partner, RXR, and we actually have good indications that that is the case. So by having these data early on, that really helped us, because we started to think about alternatives. What we're trying to do, and this is still quite a bit of a challenge, is to see if we, within this group of patient mutations, if we can find some common profiles, or maybe I should say some common defect. So we tested quite a few, and again, I don't expect you to see any of it because it is really, really small. But this is turning out to be quite difficult, and one of the reasons is that is really a technical one. When we express these mutant proteins, it's very difficult to get the levels really equal, and that might affect a lot of our data. So we're a little bit careful there. We need to repeat some of this. Also, what we included here, what I didn't mention, is that there are maybe 3-4 mutations reported, also in PPAR mutations reported in colon tumors. It's very difficult at the moment, I think, to say what is, if it is cause or consequence. I think it's probably consequence, but anyway, we did take them along. They're also in here, just to see what happens there. And of course, what we would like to do is to combine these data with expression data. So when we shoot these receptors into cells that are normally negative, and then see which genes are being upregulated or downregulated. And we're also combining this with ChIP-seq profiling together with the lab of Susanna Mandrup in Denmark to see if we can see something about DNA binding in that respect. But that's still far away. We're not there yet. And hopefully, by doing that, what we can do is identify critical nodes, and that can be a cofactor, but it could also be a target gene that tell us a little bit about maybe like the molecularly, from a molecular point of view, the minimal disease, if you want, right? What is either the cofactor or the cofactor interaction or a subset of target genes that are all linked that maybe help you to identify cases like this, and maybe you can use that in the long run, obviously not for these very rare patients, but they will tell you where to go. So it all has to do with translating fundamental findings into therapy, and what I've tried to convey to you is that to using this what I think is a very strong combination of these rare patients with a lot of molecular biology, also some modeling that I've not alluded to, and together with the MARCONI technology will help us to understand more about this, as you can see, very complex molecule sitting on DNA and driving very essential cellular and metabolic and other physiological processes. We think that this will help us to define the pathological mechanisms. Of course, it can mean that you are able to treat these rare individual patients better, because if you have a receptor that is really completely dead, I mean, it's not going to work to have a drug hitting that molecule. That's never going to give you any benefit. It hopefully will help us to identify these critical processes or critical nodes that maybe are relevant for the total population, much more than only for this small group, and also to develop new generations of drugs, obviously. And with that, I would just like to acknowledge some of the people involved. And I'm sure I put here many others because I'm sure I'm forgetting people. People from my own lab, mainly Arjen Koppen, who has really been instrumental in doing this. The modeling group led by Alexandre Bonvin in Utrecht. Of course, we need these clinicians to provide us with these rare patients. And it's really funny whenever I talk to clinicians who might see these patients and I describe what they look like. Very often they say, oh, I know a woman this and this, or I know a sister, and they look exactly like what you describe. And then, of course, then we start testing again, and that's how we sometimes pick up these patients. And, of course, the team here from PAMGene, and I really have to acknowledge here René Houtman, who is really, right from the start, has been one of the driving forces behind all of this. Thank you for your attention.